Hello, everybody, and welcome back to Creation Myths. I am thrilled tonight to have with me my guest, Dr. Fazale Rana. Dr. Rana has a BS in chemistry from West Virginia State University. He has a PhD in chemistry with an emphasis in biochemistry from Ohio University. And as of tomorrow, he is the president and CEO of Reasons to Believe. Dr. Rana, thank you so much for being here. You're have a new job starting and you're about to go on vacation. So I really appreciate you taking the time to work around my schedule to make this happen. Please take a few minutes, introduce yourself. And we're going to be talking about junk DNA, everybody, which is a really fun topic. So Dr. Rana, if you also want to just give us a couple minutes of your thoughts and then we'll just get right into it.
Yeah. Well, first of all, thanks for having me. I'm honored. I look forward to these kind of conversations. They can be challenging, but this is a great way, for me to really identify, weaknesses in my position and places where we need to spend some time doing, more work. So I love to have these conversations. So I'm thrilled. I'm honored. I've been looking forward to this. So thanks a lot for having me. And, I as you said, work for an organization called Reasons to Believe. And I hold to a position known as Old Earth Creationism, where I, in a nutshell, accept the scientific dates for the age of the universe, the age of the Earth, the antiquity of life on Earth. I believe the fossil record is a real proxy for the history of life on Earth. But I also see evidence for design in the created order as well, or in the natural order as well. And so I think that there are, certain points in life's history where there was an intervention event on the part of a creator to bring about, transformations or bring about biological innovation. I like to think about things systematically. And what I mean by that isn't that I think about things, in a sequence, but rather I like to think about things from a systems standpoint. And so, I became convinced personally that there had to be a mind behind life itself. There had to be a creator. When I was a graduate student, I was an agnostic, had no real interest in religious things. And it was just studying biochemistry and becoming aware of what are intractable problems with the origin of life that convinced me at least life's origin, again, required the work of a mind. And I've spent, that was 35 years ago or so, and I've spent the last 3.5 decades continually revisiting that conclusion and written a number of books dealing specifically with the origin of life question. So if there's an area of expertise that I would claim, that would be the area of expertise. And when I look at the history of life on Earth, again, I just see these key transitions in life's history where it doesn't look like current evolutionary theory is sufficient to account for those events. Something I'm becoming increasingly interested in is the origin of eukaryotic cells. That's a fascinating area of science and investigation. And I think that's another place where superficially you've got seemingly a compelling explanation in the endosymbiont hypothesis. But when you start digging into the details, there seems to me to be some very significant issues. And again, I don't want to get into derail the conversation.
I was just thinking I should have invited you on to talk about that. I didn't know that was one of your things. I mean, we could have done an hour on.
I don't know that I would say that I've got much mastery, but it's a fascinating area that I'm just really beginning to dig into. But love to talk about it, at some point. But but the whole point is that again, I do see. I wouldn't say that I'm an anti-evolutionist. I think there's aspects of the evolutionary paradigm that are clearly well established. There's explanatory power in the evolutionary paradigm for features in biology. It's just that I do think that there are points where a creator needs to be involved. Now, if that's the position that I hold, then when it comes to what we're going to be talking about tonight, namely shared features that we see among organisms that naturally group together, how then do I account for homology? And I I kind of tap into the thinking of Sir Richard Owen and the idea of archetypes, and try to, apply that in a more contemporary context. Could some kind of archetypical biology account for, homologies that we see, particularly at a genetic level? And if that's the case, then minimally what I have to do is show that, these shared features have some kind of function, some kind of purpose. There has to be an explanation, for example, why pseudogenes look like their corresponding genes. Why do ERVs look like viral genetic material? And then there's also the challenge of how do you account for a nested hierarchical organization of those features. So, I readily acknowledge that the case for common descent is very powerful. And so I'm not approaching it that my explanation is necessarily a better explanation, but in a design framework, can I account for those features if I choose to look at biology through the lens of design? So it's not kind of pitting one explanation against the other necessarily, but rather, how can I account for those features that otherwise seem to make a compelling case for common descent? So anyway, that's kind of my project. That's really my interest in, the whole question of, quote unquote junk DNA. And, and of course the Encode Project is rather intriguing, right?
Right.
In terms of at least the claims that came out of the 2012 paper, which was kind of summarizing the phase 2 results, which was 80% of the human genome seems to be biochemically active, which
Right.
Could be understood as being a functional significance. So, so again, highly intrigued by that result, which seems to be at least to go a long way towards satisfying, one of the criteria that I've got to, demonstrate. So.
Yep. Yeah. So to just kind of frame this for everybody that's watching, all 12 of you, however many there are, I don't know how many are watching right now. for everybody that's here, finding this just super thrilling, I genuinely am. Junk DNA, and you can even put that in air quotes if you want. You can do it either way you want, as you prefer. Junk DNA has become like this battlefield in this as like a proxy battle for this design versus not design argument that's been going on for decades in its kind of modern form. And it's the idea is that old Earth creationists, design proponents, young Earth creationists, whatever, all kind of have arrived at this position where, and Dr. I'm explaining this for the audience, just contextualizing for the audience. I know I don't have to explain this to you, that have arrived at a position, as Dr. Rana just explained, where it is consistent with their hypothesis to have a genome that is largely, if not entirely, functional. On the other side of that, you have the evolutionary hypothesis, which is looking at things like conservation and shows that there's not that much long distance sequence conservation, looking at things like mutation accumulation and the challenge that poses for the long-term stability of genomes, and seeing that, well, it sure looks like most of the genome either isn't or shouldn't be having some specific function associated with it. And for my money, I think that case has been overstated on both sides there. I think the range of outcomes that are compatible either way is much larger than either side largely acknowledges, but that's neither here or there for tonight, I think. But it largely comes down to, this argument has largely come down to basically what percentage of the genome actually has a specific function. So I was going to ask you, just how do you describe yourself in your position? Did a great job summarizing the old Earth model and exactly why this matters in that context. So my next question, real simple, and then we'll get to a really complicated question, but next question, real simple, is if you had to put a ballpark number on it, what percent of the human genome is functional, however you define function?
Well, that's not that simple of a question. I guess my answer to that question And my wife always says I never actually answer the question that's
Yeah,
posed
it's
to me.
okay. We don't
I
have to
talk,
answer
but I
this.
don't answer the question. So I'm going to do that right now. But and my wife's over here smiling. So, but I think we are quite, to be humble about it, scientifically humble. We really are in our infancy in terms of understanding genomes of organisms, let alone the human genome. And it'd be great to say, I think, probably 85 to 90% of the genome is functional. But I mean, truth be told, I don't think anybody really has enough of a mastery of understanding of the human genome to really put any kind of genuine figure on that. Right. And of course, this again depends on what we're going to be talking about.
Right.
What do you mean by
the next
function
question.
too? Right.
Right.
And so, from an old Earth creationist position, I would have a conviction that most of the genome is functional. It can't all be functional. Most of it's functional. But I think, truth be told, I don't know how anybody can truly answer that question.
Okay, so I've got you to 50.1% to 99.9%. Somewhere in there is where we've narrowed it down. That's cool. I'll take it.
So how would you answer that? How would you answer
that? I was hoping you would ask. It was if you made me go through and tally up the different parts of the genome and what I think have really strong evidence of function for, I think we can arrive at somewhere between 10 and on the outside, 20, maybe you can get higher than that if you're really generous about a few things. Like there's some ambiguous stuff like spacer DNA, where like we know that two things need to be separated by a certain amount. The sequence doesn't matter, but we know if this region is, if it's 1,000 base pairs, it's fine. If it's 500 is too short. If it's 5,000, it's too long. So if you're really generous, I think, about stuff like that, I think you could get the functional fraction into the low double digits, I think is where we have right now, I think where we have reasonably strong evidence for. And then I think leading into our next question, as you said, with the Encode project, the initial findings of the phase 2 of it were 80% some biochemical activity. And there's a number of categories of that. And so you've got that as a high end, and some of the people from that project at the time, and this was in 2012, saying, and we expect it to go to close to 100 subsequently, which in their follow-up work is not what ended up happening, but that was what a couple of the people said at the time. So you've got 80 as kind of your kind of high watermark of like what kind of the mainstream biology community, to the extent you could call Encode, I think it's fair to say Encode was part of the mainstream biology. I think that's fair. So if you call that 80% on the really super high end, I think we have really good evidence for 10% and like pretty okay for up to 20, I think is what you could, I think that's what you could argue. But I'm sure I think that's what you could argue, but let's find out, right? So the obvious question here that everyone's probably wondering is, well, okay, you disagree on this, but how are you each defining function? Because if you're defining it differently, then you may not be saying different things, right? You're looking at the same data and just drawing a line around a different part of it and calling it functional. So let's talk about the definition of function. How should we define function in the genome?
Yeah. Well, and there's, some fascinating articles that I've read over the years, that really kind of debate what is meant by function, and it becomes actually a very complex and technical conversation, which is but in some respects you could argue that this is somewhat of a philosophical discussion as much as a scientific discussion, because I think in most areas of science, when you think about function, it's connected to cause and effect. And so one definition of function would be the causal definition of function, where, as a biochemist, you study the interactions between, molecular systems inside the cell. And so, is there an interaction that takes place, and does that interaction have consequences for the cell? And that's, how I would define function in a general sense. And so when you looked at what the Encode project was doing, it was a collection of biochemical assays, looking for transcription you know, transcription factor binding, other types of protein binding to DNA, histone binding, histone modifications, three-dimensional interactions. The latest Encode release phase 3 looked at actually RNA binding as well, which was interesting. And so these are biochemical assays. And so, I think by definition, you're going to report the results and look at the results, kind of from a causal standpoint, where, are there interactions that are taking place, and do these interactions seemingly have consequences? Now, one of the criticisms that I think is interesting of the Encode results, well, actually two of them. One is, what is the sensitivity of the assays, right? Because, and I don't have firsthand experience with many of these techniques that they're using, so I don't have that wherewithal to really go in and assess, how sensitive are these methods, and is that sensitivity at the level where you're detecting noise? Although apparently they have done, they've gone out of their way to do replicates to ensure that they're producing and generating and reporting high quality results. So there are these quality control checks that they're employing. So you'd like to think that these aren't spurious results.
If I could just jump in for 1 second and say, please. I think Encode, I have some issues with the Encode work. We'll talk about it. But one of them is not that they're reporting things that are kind of background noise in the assays themselves. I think the activity that Encode reports is genuine biochemical activity. I think especially in their subsequent work, 2012 is the 2012 paper is the one that's the famous one. Everyone knows that one, but everybody should read the later Encode work where they really dig deep into some of this stuff. And they, for example, tell you like, not just was there transcription, but they go into like how many transcripts per cell, right? And like that kind of thing is really important because it gives you confidence that they're not just measuring, it's not error in the techniques themselves. It's not like they're, you talk about like error with like background noise with like radiometric dating, for example, where you have a range for each technique that works. And outside of that range, there's a margin of error where you're just operating within the margin of error and you can't actually get reliable results. It's the same thing with biochemical assays like these. But I think Encode did and does a really good job of making sure that's not what they're reporting. So I think that's totally correct. Yeah.
Right. Well, and then, I think the other criticism is just because you're measuring activity, is that necessarily functional? Now, from a causal effect definition right now, that, and again, I think that's a fair point is, these are chemical systems, they're noisy, right? And again, replicating the results is one way you kind of ensure that it's not just simply noise, but to me as a biochemist, if you start talking about large scale, let's just say transcription factor binding, where the binding is non-functional binding, non-specific binding, there's got to be some of that's happening. But to me, intuitively as a biochemist, I would say if you start having that high levels of non-specific binding, you are, first of all, you're titrating out transcription factors that are in that by titrating them out, in the system, you're not going to be able to get the functional binding you need with those transcription factors. It's kind of like a dilution effect. Secondly, if there's this random binding, that's just going to kind of gum up the operations. And so to me as a biochemist, I would think intuitively, most of that binding has got to be serving some kind of purpose. Or if the transcription is just the cell's just transcribing DNA without it having any purpose, that again creates a very messy environment in the cell that would gum up the operations, and it's energy expensive. Right. So these things, to me, intuitively as a biochemist, seem to argue against that. This is just this is non-specific activity that you might call noise, right?
I absolutely hear you. So let me tell you my definition of function, because it's a little bit different. So my definition of function, and this is not something that like this is my headcanon, this is how I define it, is it's so I want to make sure I have yours correct before I state mine. I want to make sure we're all on the same page. So it's interaction that has consequences for the cell, biochemical interactions that have consequences for the cell. It's the causal definition of function. So mine is a little bit more restrictive, and this is why we come down with different ranges in terms of what that functional fraction is, because mine is activity, biochemical activity that affirmatively contributes to cell physiology or organism physiology in some way, by which I mean there is some benefit to, like, it serves a purpose for the functioning, the physiology, something going on in the cell. And it's not just, not 100% perfectly gated transcription regulation, for example. It's like there's some leakage there of low-level spurious transcription that happens. There's protein binding that happens. There's DNA modification that doesn't do anything except tell that part of the DNA to shut up and sit in the corner of the nucleus and don't do anything. Like, so that tells you that, like, that's an interaction, right? But that part of the DNA, it's not doing anything for the cell. It's just like maybe you methylated a bunch so you can't do transcription initiation, and then it just sits in the corner and doesn't do anything because it's super dense heterochromatin. If you ask me, that I think would count as functional under the causal definition of function, but under the kind of, I don't know how you would describe mine, because it's not strictly speaking the evolutionary definition of function, even though I do like to consider conservation as part of the equation. But under my definition, then that type of interaction, the DNA that's involved in that, even though the process is functional, right? Targeting a specific region for methylation, that's functional. But the DNA sequence that's affected by that, which then sits densely as heterochromatin somewhere so it doesn't get in the way, that DNA is non-functional. Does that distinction make sense?
It does. It does. And it actually isn't, it probably wouldn't be that, there's some overlap, I think, in terms of the way that we're thinking about function. Right? I don't think it's, I don't think the positions are necessarily mutually exclusive. And I mean, and again, it depends on, I guess, how you think about, contributions. So, you were making the point that, I mean, it's interesting to me as a biochemist that we're now thinking about genomes as three-dimensional entities, right? Not these two-dimensional or one-dimensional. Right? But really is three-dimensional entities, which means you may have sequences of DNA that are serving no other purpose other than to help to get, elements at different locations in the genome in the same three-dimensional, location. And so you could argue that again is function. Or an intriguing idea, I think maybe is it W4 Doolittle suggested is years ago is something called the nucleoskeletal hypothesis, where the argument is that DNA is actually helping to regulate the volume of the nucleus, and that the nuclear volume, the ratio of the nuclear volume to the overall cell volume, is a very tightly controlled parameter. It can only, it's a fine-tuned parameter, if you will, and so the amount of DNA actually is controlling the nuclear volume, and so you could argue that that's function, in some respects, or could some of the DNA even just serve as a mutational buffer, right? Where
that's a popular hypothesis, yeah.
And that kind of gets to some of the arguments that I've seen for why the genome can't have more than about 25% functional elements is because otherwise it wouldn't be able to tolerate mutational load. So you could argue that's a type of function, right?
Right.
I'm not.
Right.
I would say I would agree with that, but I think there's the elements that are functional in the genome are doing other things beyond that. But
okay, yeah. So that's a road we could go down and probably spend an hour just on that, because there's the famous Grauer paper about the functional fraction, and the famous, if Encode is right, evolution is wrong, which is not what he meant, but that's what everyone says he meant. And then there was the revision to the paper in 2020, and then he put a correction on the original paper. It's a whole thing, and everyone just references the first paper and leaves out the whole rest of the story. And everyone watching, we're not going to go down that rabbit hole tonight because it's to the side of what we're talking about, but there is a whole story there, and it's a fun story. But that's neither here nor there. So this gets into something, what you just described gets into something really interesting. And one thing that I think needs to be considered when we think about this stuff, because again, I've read your writing on this. And you're talking about the causal versus the evolutionary view of this. And I'm an evolutionary biologist, so I come to this from an evolutionary perspective. And I look at something, and if I see that there's a sequence that, it's a retrotransposon, I can, there's mobilizable elements that are dead on matches to this thing that's mutated in our genome. The one in our genome, it's not really doing anything, but it sure looks like it was ancestrally a retrotransposon. And it turns out, though, actually, you know what? Retrotransposon's a bad example. Let me use pseudogenes as an example instead, because you mentioned pseudogenes earlier, where you have a pseudogene that's, kind of an inactive version of an active protein encoding gene. And the pseudogene, it's mutated. Maybe it's got a premature stop codon, or it's missing a promoter or something. It's missing some parts of it, right? And but what we find is that in cases where pseudogenes are transcribed, sometimes they're affecting the translation of the actual mRNA, either as kind of like decoy targets for small interfering RNAs, or they could actually downregulate the actual mRNA in some way. And the way I think about that is because looking at pseudogenes, it's clear, they're not under purifying selection, right? They can mutate, and there's no apparent fitness cost to those pseudogenes being degraded or deleted or anything. So that tells me, like, okay, that sequence, right, within that region that we call a pseudogene, that sequence itself doesn't actually matter for whatever it may or may not be doing. So the way I think about stuff like that is, if you could think about, you got a house, I have an old house, so like my house is not like, the walls aren't insulated everywhere. So like in some rooms in my house in the winter, if I want it to be 72 degrees in the room, I have to set the thermostat to 74, right? Are my drafty walls helping regulate the temperature of that room? Yeah. But are they like serving a purpose in regulating the temperature of that room? No, it's just affecting it, and I kind of have to compensate for it in some way. And that's how I see a lot of this stuff, where we have a lot of the genome that's derived from transposable elements, and that affects the 3D structuring. We have pseudogenes and other transcripts that may affect the level of gene expression. Are they regulating it, or are we kind of just dealing with the fact that these processes are biochemically messy and imprecise, and we have to, kind of up or down regulate in response to that to get the outcome we want, through other means? That's where that kind of stuff gets fuzzy for me, because it looks like this stuff isn't constrained to do the job that it does. So it's more interfering than regulating.
Right. Well, and you're bringing up the topic of pseudogenes, and again, I'm approaching this from the standpoint that we're still learning. Again, our understanding is in its infancy, and that things are developing, right? And again, in light of the project that I'm engaged in, something that I find fascinating is this idea of the competitive endogenous RNA hypothesis. And I am of the opinion that this very well could be an idea that becomes kind of a companion to the central dogma of molecular biology. That's my intuition, and I could be wrong, but that's my intuition, and I'm beginning to see more and more evidence in support of this idea, which is essentially arguing that in the 3' untranslated region of messenger RNAs, there are these microRNA recognition elements, and that those MREs actually will bind specific microRNAs. And so you'll get families of messenger RNA transcripts that are all binding the same MRE, which then targets the messenger RNA for degradation. And so the idea is that, when you start affecting the level of one transcript, it affects the entire level of transcripts in that particular family. And so it starts to provide what I would dare say an elegant explanation for why, pseudogenes look like, quote unquote, the intact versions of genes, or the healthy versions of genes, however you want to describe it, right? Because now you have kind of like a decoy effect going on, where the pseudogene expression actually is helping to regulate the expression of the intact gene in ultimately in the translation of that gene. And and yet again, the fact that there's these sequence similarities are necessary, for it to function in a decoy capacity. Long non-coding RNAs also seemingly enter into these transcription networks. And so this is an example of where the kind of the concepts that you're saying can be pushed forward one step further to say this seems to be a much more elegant system than just something that would have been kind of cobbled together, if you will, as a result of an evolutionary history. There seems to be an elegance to this, and so these are the types of advances that I think are intriguing. Again, there seems to be this underlying rationale, dare I say, that seems to be accounting for, again, the presence of at least duplicated pseudogenes in genomes. And in fact, you could even argue that unitary pseudogenes, are still playing a functional role in gene regulation because they are part of this transcription family, if you will.
So there was one thing in there that you said that I think is really important to this whole conversation. And so we have pseudogenes, and they have all this apparent biochemical activity. But the really critical thing that we see in there is that they are not conserved. They are not under purifying selection. So what we can see with these sequences is that they can have deletions, they can have mutations that make them different from the paired pseudogene gene kind of pairs. The pseudogene can experience mutations that make it diverge from the parent gene, if you will. And there's no apparent cost to that, despite the role that they may play or the influence they may have in affecting the level of translation. And this is important for a lot of these different elements in the genome. And you can go kind of top to bottom through endogenous retroviruses and signs and lines and all these things. And you could see, as you described, really compelling examples of biochemical activity that seem to be really important for the cell. But then you look in all human genomes, for example, and where you find single nucleotide variants. You look across all primate genomes or all mammalian genomes. And what you end up seeing is an extremely low level of conservation, where within mammals, it's like 5%, I think. You get strong purifying selection. The rest is approximately neutral or deleterious, or not deleterious, rather neutral, or it kind of depends. Once it changes, and then it can become harmful. But it's when it reactivates, it's weird. We can talk about that separately because that's a whole other question. And then within primates or humans, you might have lineage specific regions that are more constrained that get you up into the 10% range for conservation, where it's under purifying selection. And if these pseudogenes, for example, are doing this sequence dependent activity where it's almost like Müllerian mimicry, but for microRNAs targeting the transcripts, if everyone looks the same, then you all spread it out a little bit. And if they're actually doing that and it's sequence dependent and it's critical for function, then it should be under purifying selection, right? There should be a cost to disrupting that activity. But because we can see, just in humans, we can see lots of SNP variation, single nucleotide polymorphisms in these sequences with no apparent cost, that to me seems to undermine that idea that it is a sequence-dependent, critical regulatory activity. And this also works for endogenous retroviruses and the idea that they interfere with actual viruses. Well, that has to be a sequence-dependent interaction, but ERVs are not under purifying selection, except for the couple that have been co-opted. So that's why I don't find that argument compelling, because it's not dependent on sequence specificity, and it should, that biochemical activity should be. Does that make sense?
Yes, it does. And actually, I appreciate that insight. That's actually helpful to me because it's something that I need to go and think about some more. but and let me ask you this question, and I'm asking now more from a curiosity standpoint than anything else. So the way in which you're determining purifying selection would be through sequence conservation from organism to organism, or are you looking at like the ratio of synonymous to non-synonymous mutations?
So I think you have to look at it in a multifaceted way. For a lot of these sequences, you can't do the DNDS ratio, the synonymous to non-synonymous ratio. For everyone watching, so what this is within a sequence, you could have synonymous mutations. These are mutations that don't change the amino acid sequence, right? They're synonymous. So the codons change, but what it reads to doesn't change. And then non-synonymous changes are ones that change the amino acid. Under purifying selection to stay the same, you have more synonymous changes than non-synonymous because changes are selected again. So you have a skewed ratio of synonymous to non-synonymous. Now, there's a recent paper on that I think you might be alluding to there that changed the math on that a little bit. But the sequences we're mostly talking about here are not protein coding. So DNDS ratios shouldn't matter. For things like pseudogenes and transposons, because it's not protein coding, so like the codons don't actually matter. It's not, we don't have to worry about that. Sorry, was there second part to that question? That's the idea, is that it does. So there's conservation, right? And then if it's in a non-human thing, you could actually just do experimentally, you could delete it, you could silence it in some way. There's different ways of doing that. And you could evaluate, like, is there a fitness cost to this, whatever it is. And you could do this. I don't know of anybody that does this, but in my, fantasy land, like someone says, hey, here's $10 million, do whatever you want with it. One thing I would really love to look into are these unicellular eukaryotes that have these enormous genomes, an order of magnitude or more larger than the human genome, and just like delete huge parts of it and just see what happens. Like, do they need to have that huge genome? Or can they get away with the genome that's half the size or a tenth the size and still basically work okay? And we don't actually know the answer to that question, to my knowledge, because we're mostly doing the stuff with like multicellular eukaryotes, which have smaller genomes, oddly enough, than the unicellular ones. But so you have to, I think conservation is one way to look at it. I don't think DNDS ratios are good for most of it because, again, it's non-coding. And then you could do like knockout and silencing kind of stuff. And the fourth part of it is, finally got through the whole answer. The fourth part of it is just look at the specific activity you're talking about, right? So again, one thing that Encode talks about a lot is methylation targets and histone modification and like chromatin modeling. So is it persistently heterochromatin that's really dense, or is it more diffuse euchromatin? And I think that's a really important question when it comes to function, because that heterochromatin, once it gets super condensed like that, enzymes just can't get in there and like do stuff with it. So it can be a really strong site-specific target for that type of either histone or direct DNA modification. But then once it's been modified like that, that's going to be really strong activity. And it's probably going to be conserved. Now, the sequences that are doing that are short. They're tiny little target sequences, so it's not a big percentage, but they are specific sequences that need to be conserved to do that. But once you do it to a region, then it just sits there. It's not doing anything. It's the box you bury in your basement and then pile a bunch of other stuff on top of it and like maybe kind of use like the DNA nucleus spatial analogy. It's like the box of stuff in your basement that becomes like a makeshift shelf for the stuff you actually want to be able to get down when you need it. So I think that's the answer there, is that I think it's an unsatisfying answer, because the answer is there is no one standard I like to use. I think you have to look at multiple different things and kind of synthesize it on a case-by-case basis, which not a satisfying answer because it's not simple. You can't look at something and be like, yeah, that's totally not functional, or yeah, that definitely is. It's not that easy.
Yeah. And no, I appreciate that. That's actually helpful to me a lot. And it just seems to me, and again, there's no data that supports this necessarily, but it seems to me that we ought to be cautious about simply saying that the sequence changes that we see, for example, in like a duplicated pseudogene aren't consequential, right? Because the RNA secondary and tertiary structure may be very important in terms of how the RNA is being involved in these regulatory networks. And so again, there's not, this is a biochemical intuition more so than based on, that there's data, definitely showing this. But, I think you're making a strong case or a strong point. But again, I still think we are just so much in our infancy that, there's going to be things that are going to surprise all of us that I think we're going to learn about, about the functioning, of the human genome.
I hear what you're saying, and if I can see if I understand your point correctly, you could have, for example, so you have an actual true gene, protein encoding, transcribed, translated, whole 9 yards. You've got a duplicated pseudogene, kind of broken down, but the sequences still more or less match. And it's not necessarily the sequence in that pseudogene that's doing something, but maybe there's something about the secondary structure of that RNA that's doing something. And you know what? Maybe you could have a C to T substitution over here, and but you have an A to G over here, and it preserves a particular stem loop. So there isn't going to be constraint at the sequence level as long as you have but you do have constraints at kind of the secondary structural level, and as long as you're within kind of a range of the secondary structure, that mRNA can accomplish whatever it's going to do. Even though it has kind of the freedom to mutate in different ways. Is that a fair representation?
Yeah. And again, I'm, it's just simply saying that there are ways I think you could accommodate, the idea that the sequence doesn't seem to be under purifying selection, when in fact, it still could be. There could be a tolerance for mutation, or maybe some of those changes actually are of functional consequence. Where that duplicated pseudogene is serving as a decoy, and the sequence similarity with the intact gene is sufficient for it to function as a decoy, but as it's entering into this network, secondary, tertiary structural features may influence the way in which it's degraded. And that could impact what's happening with the overall family of transcripts. So there's, I think, several different ways to think about that, where your point doesn't necessarily obviate,
there's levels of function independent of sequence specificity that we may be missing, because the assays that we've been using, even including Encode's assays, are predicated on sequence specificity. Because those are the interactions we know about just historically in biochemistry. They're whether they're amino acid interactions or nucleotide interactions, they tend to be relatively specific. I think that's a totally fair point. I think that's fair. I don't think the data are there yet to be able to assign a lot of function through that mechanism, but I would not say it's impossible for that to be the case. I think that's fair. So we've already been going for almost 45 minutes, which
it just feels like we started.
I'm like, but I do. So I think this is actually a good time, because to kind of not necessarily wrap it up, but try to converge on some kind of pseudo conclusion here, if we can. So let me, I wrote out kind of, as best I could. I mentioned off air that, every time you go year by year looking at the pie charts of the genome, and they're different every year in terms of what's what. But I put together kind of a rough approximation of what's what, kind of in different bins by classes. And if it's okay with you, I'll just go through the percentages, and we could just say okay, this is this is that, and kind of just see where we land on stuff. Does that sound okay?
Yeah.
Let's just do this. So I have, and this is some of these numbers are precise because they're papers that have we're going to find out the specific fraction that does this thing. And some of the numbers are not precise, and they're just it's about this much of the genome does this. You're okay. So looking at stuff that I think we can agree is functional. You've got exons, obviously. Exons are functional, right? Protein coding, simple. RNA genes, exons are a percentage, 1 to 2% of the genome. RNA genes, if you're really generous, you can get to 3%. You got ribosomal RNA, tRNA, you've got your microRNAs, you've got a bunch of other classes of RNAs, some long non-coding RNAs that have like documented functions. That gets you like another like 3%. You've got your enhancers and silencers. If you're really generous, you can call that 1%. It's not really 1%, but like you round up to 1% for that stuff. You get your origins of replication, telomeres, centromeres, and telomeres and centromeres are really interesting because they're not, it's, the sequences matter, but it's just like repetitive stuff that says you're in the middle, and then there's more repetitive stuff that says you're at the end. And then you've got your spacer DNA that I alluded to earlier, which is not sequence specific, but the size is constrained. And if you mess with the size too much, it causes problems. And if you add all that stuff up, like the centromeres like 5%, telomeres are tiny, they're less than 1%, origins of replication are like 0.5%. You add all that stuff up, you get like 16% of the genome. And that's being pretty generous with some of the estimations. And I think we can agree on that 16%.
Yeah.
That's all functional. That's cool. Then you get the rest of the genome, and that's where it gets interesting because you've got your 5% of the genome, give or take, is pseudogenes. Some of them are transcribed and match actual genes to one extent or another, but some of them are like not. They don't have promoters. They just kind of sit there. Could they be doing, like, what about a pseudogene that doesn't have a promoter anymore? It's like missing, promoter, exon, and then like the rest of the gene is kind of there. Could that have a function?
It possibly.
I mean, obviously it could, but like, what's the hypothesis for that thing to have a function?
Right. Oops. Yeah. Well, there was a paper. I ended up writing a blog about it a couple of years ago now, maybe, basically pointing out that the level of transcription among pseudogenes may actually be higher than what we think, simply because the assays that are performed many times might actually conflate pseudogenes with the intact genes. In other words, the pseudogenes are being transcribed, but it's being reported as being the intact gene.
Right. It's hard to distinguish between the two.
Right,
yeah.
Right. Mm-hmm. And again, if they're not being transcribed, that's a problem for my model, right? Mm-hmm. Unless they're serving some other role, again, that hasn't yet been defined. Mm-hmm. I think in this particular paper, and I'm going from memory, so there was some comment to the effect that maybe these untranscribed region or pseudogenes may be playing some kind of role in chromosomal stability. Now, mm-hmm. I can't remember how they specifically were making that claim, so I'm going from memory. I probably should have reviewed that before this.
Oh, that's okay. Yeah.
But so perhaps they could have some kind of function.
Okay.
But I would agree that kind of counts against my model right now.
Mm-hmm. So the biggest class that I have in my notes is retrotransposons, which there's a bunch of subclasses, but you put them all together, it's like 40% of the genome are these retro elements, where it's, for everyone, retrotransposons are things where they do this copy and paste thing, where it's a little bit of RNA, it reverse transcribes the DNA, puts itself in your genome, and then it transcribes back out, and then reverse transcribes and puts itself back in somewhere else. So you end up with these copy paste things where they just end up copying themselves all over the genome. Now, there's a bunch of different categories here. There's your lines, your long interspersed nuclear elements, signs, short interspersed nuclear elements, your LTR retrotransposons, which look like endogenous retroviruses, but they're missing a part, so they're different. That all told, it's like 40 to 42% of the genome. I know that we have, so there's some hypotheses with like gene expression and alternative splicing with some signs. There's the main class of lines, the L1 lines, the L1 retrotransposons. That alone is like 17% of our genome. It's huge. Some of those they affect gene expression patterns. Where do you come down on stuff like that? These retrotransposon derived elements that many of them are degraded, and if they're transcribed, it's at very low levels, and many of them are not. So where do you come down on those?
Yeah, and I have not, made any attempt to say this particular percentage seems to be functional versus, I'm
going to hold you to it. It's 50.1 to 99.9. I'm going to.
Yeah. But again, but I would see those kinds of discoveries of, line in sign DNA playing regulatory roles, the LTRs playing regulatory roles, as suggestive of the fact that maybe these elements are doing things that we just simply are not yet aware of.
Mhm.
And and part of my model would be, how do you then explain the similarities of these sequences from, retroviruses that they presumably have been derived from? Is there something about the similarity in the sequences that makes elegant sense in a design framework, why
Mhm.
there would be similarities? And so, for example, with endogenous retroviruses, at least the particular classes that are expressed, you could argue, and there's papers demonstrating that they're playing an antiviral role.
Mhm.
They're providing a kind of an innate immunity against, viral infection. And it's not just simply retroviral infections, but viral infections in general. And so, I think that's intriguing. There's some recent work about, the expression of the, I can't remember the class of ERVs now off the top of my head. Is it the HERVK
Yeah, it's the
family?
Ks or is that the
Yeah.
cell fusion that you're referencing?
No, it's not cell fusion. It's essentially that they're expressed at very high levels during the very early stages of embryonic development,
Yes.
when the embryo is vulnerable, and that this is providing kind of an antiviral defense.
Mhm.
And then as soon as you hit a certain stage in development, and it's like the SOX2 gene, which is essentially turning on this ERV expression, and that's also keeping the blastomeres kind of in a pluripotent state. And as soon as differentiation starts, SOX2 expression is
Mhm.
shut down, the ERVs are silenced. And so, to me, that's a really elegant system that is suggestive that there may be things going on with ERV sequences that just simply haven't, been discovered yet. So that's how I would view that, is that
Yeah.
it's promising, and that there's a rationale for why the ERV sequences would look like retroviral sequences.
So this gets at something that I'm glad you brought that up, because this gets at something that I think I've read from you. You've said that we have strong evidence of I'm paraphrasing. Correct me if this is not an accurate representation, but I think you've written that we have strong evidence of function in every major
Right.
class of DNA element, where you could go
Right.
herbs, signs, lines, whatever. And you could say, here's an example that we have really strong evidence does a particular function and is selected to do that function.
Right.
And I think you're absolutely right when you say that we have strong evidence of function in every class of DNA. But where I get off the train is, okay, we have evidence of some members of that class, but if a
Right.
class of DNA, endogenous retroviruses are a great example, because that's 8% of the genome. There's like 200,000 of them in the human genome, but only about 20 are the full, like, LTR, gene, LTR, like the complete structure. And the rest are degraded in some way. And most of them are either just the paired LTRs or just solo LTRs. Many of them, if they're transcribed at all, it's very little. And most of them aren't. They're just methylated and go and sit in the corner and don't do anything. And so you could have a situation where you have retroviruses that have been co-opted for this very important function. And I think there's super strong evidence that without retroviral endogenization, mammals would not have evolved the way we did. I think there's super strong evidence for that. But I don't think you could extrapolate that to most of that 8% of the genome that's retroviruses, just because the ones that are doing the activity for which we have, I think we would agree that they're functional. The ones that are doing that don't look like 99% of those sequences in the genome. And the rest of them, it's not just that we don't have evidence that they don't do that thing. It's that structurally they can't be doing that thing. They just don't have the parts to do
Right.
it. And I think that concern works across the board when we talk about like ALU elements. Well, okay, there's some of them that are, in exons and are clearly have been co-opted as part of the actual coding sequence of a gene. But then most of the rest aren't actually transcribed at very high levels at all, if they're transcribed at all. So again, they're not doing that thing and they're not translated. So like, that's where you could argue, yes, there is, I think, strong evidence of function in every class of DNA, but extrapolating that to a majority of the DNA sequence as a whole is challenging.
Yeah, and I think that's a fair point. And that's, at this point in time, that's the, I wouldn't say that's the limitation of my particular argument. And I, and I'll say this, that when I started working at Reasons to Believe, one of the first papers I wrote was what do we do with junk DNA? Kind of,
I think I read that one actually. It was like 2005 or something. I think I read that one.
Like 2000, yeah.
Oh yeah.
And it's like, and there was really very little we could say, and so it's interesting to me that, over the span of a couple of decades, we now are actually talking about at least some ERVs having function, or duplicated pseudogenes having function, because truth be told, I'm not so sure that I could ever have imagined that being the case, through these contravening years, so to me, there's a trend line that's rather intriguing about, recognizing functionality, in the genome with regard to different sequence, classes. And so I go back to the point that I made earlier. I think we're in our infancy in our understanding. And so I would be a little bit circumspect about, at this point, throwing in the towel, even though I think your point is a strong point. I'm a little reticent to throw in the towel at this point, just because of what I've seen over the last couple of decades. And it could be that, at the end of the day, you're absolutely right. And then that would, I think, really it would really make a very strong case for kind of an evolutionary, origin to life's history, that's, clearly evidenced within the genome, but again, I'm not quite ready to throw in the towel. And I think there's a lot of interesting things to come,
I 100% agree. We absolutely should keep digging into the human genome as much as possible and figuring out what every little bit does at all. We should 100% keep doing it. There's no question about that in my mind. And there's also no question that we're going to find more stuff that has important functions. I think that's absolutely the case. I think we both think that. I think we just think where that ends up is going to settle at a different point. We're coming up on an hour, so I think that's a really good place to end this. Since we've been talking about herbs, I have one more question for you, and it's, I hope, a very brief question, and I'm going to apologize ahead of time because you don't know this, but you're going to wade into an ongoing debate that's going on. So I apologize. But and then I'll ask you if you want to just take a few minutes and summarize your perspective, and then we'll call it a night. So endogenous retroviruses, actually the result of viral endogenization, or put in place, designed in place for whatever reason? Where do you come down on that one?
From a old Earth creationist design perspective, I think a significant number of them were probably designed that way because of kind of an antiviral role, and maybe other roles that we've yet to discover. I think there are some of them that probably have been introduced through an endogenization process. So cool. To me, it's a mixture.
Okay, interesting. All right, cool. All right, so if you want to just take a couple minutes and summarize your perspective, and then we'll call it a night. I just thank you so much for being here. This was super fun, and I really hope everyone watching enjoyed this conversation as much as I did. This was really cool. Please take a couple minutes and say whatever you want to say. I just want to say
thanks. I enjoyed the conversation, and it's been helpful to me, and I appreciate the spirit behind the dialogue. So thank you for having me. And I guess at the end of the day, as someone who holds to an old Earth creationist position, I think we're going to end up discovering that the genomes of organisms are these incredibly elegant and sophisticated systems that display, design in ways that we can't even imagine. And so to me, it's an extremely exciting time to see the advances that are taking place. And so, I think that, the trend line is going to be that we're going to see more and more, function in the genomes of organisms.
All right. Well, thank you so much, everybody. This has been Dr. Fazale Rana. He's been with me. We've been talking about junk DNA and a bunch of related topics and kind of meandering a little bit kind of in that ballpark. And it's been super fun. I hope everybody enjoyed that. And Dr. Rana, thank you so much for coming on. I really appreciate you giving me so much of your time. Have a great night. And everybody, have a great night. I will be back at some point because my schedule is sporadic, but eventually I'll have more stuff. Thank you for watching, everybody. If you haven't done it already, hit the like button and share if you enjoyed this. Have a great night.